This is nucleus. Nuclear envelope enclose the nucleus, separating its content from the cytoplasm. Control the passage of various substances into and out of the nucleus. Nuclear pores, the inner and outer membranes, fuse at certain intervals, which form nuclear pores. It regulates the movement of substance across nuclear membrane.
Nucleoplasm.
Semi-fluid matrix fills the nuclear space between nuclear envelope and chromatin.
Chromatin.
Package DNA into a compact form within the nucleus, enabling efficient storage.
Nucleolus.
Spherical, surrounded by a layer of condensed chromatin, site of ribosomal RNA-rRNA synthesis.
This is endoplasmic reticulum. Structure of rough ER. Cisternae. Membrane-bound sacs or vesicle. Flattened. Cisternal space. The internal space enclosed by the ER membrane. Ribosomes. Give rough ER its rough appearance. Function of rough ER. Synthesis protein. Assemble protein. Transport protein. Modified protein. Structure of smooth ER. Membrane of smooth ER. Absence of ribosomes. Lumen. The internal space enclosed by the ER membrane. Forming a continuous network of interconnected tubules and sacs. Throughout the eukaryotic cell. Tubules. The network of interconnected tube-like structures. Forming the basic architecture of the smooth ER. Function of smooth ER. Synthesis lipids. Detoxification of drugs and poison. Storage of calcium ion. Metabolism of carbohydrates.
The Golgi body, or Golgi apparatus, is like the cell's post-office sorting, packaging, and shipping proteins and lipids. These pancake-like sacs are called cisternae, each with an internal space called the lumen. The CIS face is closest to the nucleus and endoplasmic reticulum. It receives newly synthesized proteins and lipids. The trans face is oriented toward the plasma membrane where it buds off vesicles for transport. In plant cells, Golgi body secretes polysaccharides to form cell plates and cell walls. This is lysosome. Lysosome is small, spherical sac. It has a single membrane. It contains hydrolytic enzymes. Phagocytosis. A cell engulfs a particle by extending pseudopodia around it and pinches off to form phagosome. A primary lysosome fuses with food vacuole to form secondary lysosome. A hydrolytic enzyme digests the ingested particle.
The process of autophagy.
The first process of autophagy is sequestration.
A damaged organelle is enclosed by a single membrane to form autophagosome.
Primary lysosome fuses with autophagic vacuole, forming secondary lysosome.
Hydrolytic enzyme digests the damaged organelle.
The autolysis process.
When lysosome membrane dissolves, hydrolytic enzymes are released into the cytoplasm. A hydrolytic enzyme digests the whole cell by autolysis process.
This is a vacuole. Inside a vacuole, there is a cell sap. Cell sap contains water, nutrients, carbon dioxide, and waste. The function of cell sap is to help cell maintain its pressure. This is tonoplast membrane. Tonoplast membrane is a semi-permeable membrane. Its function controls movement of molecules and ion that enter and exit the vacuole. What will happen if there is no tonoplast membrane? Plant cannot maintain its osmotic pressure. Imbalance of molecules and ions. Plant will eventually droop and die.